UK. He has worked as a scientific investigator at the Medical Research Council and the University of Cambridge Biochemistry Department. He's a member of the Board of Directors in the Metabolomics Society and a member of Cambridge Systems Biology Center, Cambridge Neuroscience and Cancer Center. He joined EMBL-EBI in October 2012 and is currently managing and coordinating Cosmos, a large EU infrastructure project on metabolomics standards. He is also leading the curation efforts of MetaboLights, the first open access and general purpose metabolomics repository. And today he will be presenting about MetaboLights and Cosmos. So without further ado, allow me to introduce Reza.
Thanks. Can you hear me all? Yes?
Yeah. Yes.
Okay, I thought I'd prepared a bit more than 30 minutes, but I would go a bit faster. Basically trying to talk about both Cosmos and Metabolight and where we actually try to provide a Metabolomics community. Now there's different aspects from capturing data in a standard way, standard format particularly, and make it available and accessible via Metabolights services. So it's quite ongoing, and I think it started in 2012 essentially. That official launching was in some June. So it's been 2-3 years in service, and a lot of time was into developing a service, and we just going into the second phase of its development by second round of 3-year funding to mainly now have more applications and more data analysis tools integrated, embedded into Metabolights. Still encouraging our community is still a lot slower in adopting some open exchange format compared to, for example, proteomics community. So we're trying to link with proteomics community in making people and within the community to adopt these formats, as well as clarifying rules. We've got data coming that requires dealing with privacy and ethics. So 3 main part is the data submission that we have to get a metabolomics data, the repository for reference compounds. This is where we get, down here is a repository for where we get a reference compound. So what metabolites has been detected, for example, with mass spectrometry or NMR, how does that individually look, as well as overall what a metabolomics experiment has. So we capture all different aspects of it. The main part is capturing all this metadata. So we use a tool called ISA Creator, standing for Investigation Study Assay. So this is a tool which from Susanna Sansone group, originally from EBI, now she is in Oxford Research Center. They develop these tools to capture metabolomics data, or any data essentially, capture metadata. But what it does is actually brings an ontology lookup, so people can actually link ontology terms with existing metadata. It has a kind of an Excel format, so familiar to biologists and runs on Java, so you can actually install and use it in different platforms, as well as linking that, essentially that metadata to the raw data. This is sort of a screenshot. What we get this configuration tool.
Reza,
narrow down.
Reza, sorry, I don't think your slides are updating. We can't see your screenshot.
Is it stuck in something else?
Well, we can see your mouse cursor, but all we see is the PowerPoint. Oh, there it is. There it is. Now we can see it.
Okay. I use, instead of main screen, I use. So how long has it been going on like this?
This is the only slide we saw besides the intro.
Okay. I think just very few. Essentially, this is the structure of ISA Creator. It creates an ISA format, which is based on a larger investigation, a high level, and then what is there in your study. For example, our case studies are metabolomics study, and in there you can have several assays. And assays are, for example, a mass spectrometry assay in negative mode, or positive mode, or NMR, or any other tools. So within that context, this is what we use this capture, but obviously the tool has more flexibility to be a multi-omics tool in capturing metadata. It provides this configuration means that we can actually define that, how to report an experiment. So we base our configuration file on what is community accepted Metabolomics standard initiative. So people have come up with a set of reporting guidelines, and this is what we implement to report. And then using lookup within that tool, for example, to look for certain ontology terms, for example, organism from NCBI and UIT, to link data to right control vocabulary. So this is a screenshot of the tool that, for example, uses search for C. elegans and then linking it into the right species. This is overview of how the package overall is. So the path of this complex that I try to quickly guide you is this path that we took for metabolite submission. The tool will be ISA creator. People will download that and start capturing data. And once it's done, it could be used to submit directly into MetaboLights. You can bundle up with all the data as well into big archives and then submit it. But depending on the size, really, it's sometimes tricky. If you've got very, very large studies of tens of gigabytes, it's not really easy to do it via web portal. So there are other means that we do and we explore. Advantage of having the metadata in that format is we can also use the other modules that they build. To, for example, for data analysis, the link to RISA or Galaxy, or most interesting part would be to convert all the data to RDF and using ISA to RDF and linked ISA. So this is mainly we're trying to have more concrete curation guidelines and policies and document these things so we have a uniform kind of reporting so we can use this RDF-based enriched metadata to actually provide new ways of looking at the data or creating new knowledge. As well as there are partner journals with ISA team that are using the same format. So people who use for scientific data journal or Giga Sciences, they can use the same format to use for submit there. This is a sort of screenshot on different configuration file, and depending on type of study they have, is it in vitro or plant or clinical, there are agreed reporting guidelines that basically we load up. But it's not limited. You can also edit, add, change, modify according to your need. And the last part will be a plugin that we develop for the tool to be able to show the metabolites that has been identified. It has a little lookup, use a PubChem, and they search for the term or metabolite they're looking up, and it finds the right match from PubChem because it's the most database with most chemical or metabolites or chemicals that it has, and we sort of have preference lookup on there to be a KEBI ID if it's available. If not, they can choose another type of ID for a database reference. And it automatically populates all other metadata, for example, SMILES and INGs and chemical formula, which are important for correctly identifying or reporting the metabolite that has been identified. The rest of the metadata is not shown, but this is sort of experimental, so users know where to import, for example, the mass to charge ratio or chemical shifts or anything related to the experimental side. Again, all the entries will be curated in KEBI. If they do exist, use. If not, we can curate new one. And this is sort of got challenges of its own because there are a lot of new metabolites that are not in KEBI. And sometimes identification is not complete. So there are often that they can, to a certain level, they can identify a class of a compound. Or for example, in terms of lipid, they know how the molecular formula is or how many carbons on there, but they can't place the double bond. So there are new entries to create all the time. Essentially, I talked about RDF part, but we are trying to use this data. So when you submit a data, I'll show that a bit later, you get an accession number. So this accession number, which will be unique to a study, starts with MTBLS and then a number. So if you see here MTBLS2, for example, is one of the early examples that we followed using linked ISA to create an RDF. So these are still not, it's challenging. We have to go through all the studies before it's too late and the number grows, becomes unmanageable, to find a way, a unified limitation on how people annotate their result, so we can actually make use of RDF into the data sets, and ideally we provide, make it into available eventually, but at the moment it's primarily testing. So once they're repackaged, at the moment we don't have, either you can submit the data via FTP or the web portal, but depending on the size, sometimes it's not manageable. So we're looking at same as other resources within EBI. They already done it to use Aspera, maybe some kind of bundling of data and metadata, and then submitting that into MetaboLights. So this is sort of MetaboLights, how it looks up. You got different browsing. You can browse studies, or you can browse compound, and there are browser species, and there is another new one which sort of analyze data, which you can't see in the main website, but in a dev and in sort of individual developers web version of it is an analysis site which we sort of gradually working on. Submission standards, you choose a date, submit, you can keep the data private for a long time. You can provide reviewer access during while the data is on the private to make it public. You can also, within EBI, try to link these data sets into a sample DB, so you can look up cross examples. So this is the first example in TBLS1 that we use that data set to sort of report that into sample DB, which is another service. What it does, and essentially, ideally, would enable to look up different multi-omics that have a common denominator being the sample, to be able to find whether there's a metabolomics data set for it, there's a transcriptomic data set for it, there's a proteomic data set for it, there's a genomic data set, and they all come from the same sample. So it would be one place to look up different services. The metadata has been visualized, and mostly study design. So if you remember the ISA, Investigation Study Assay, a lot of it is sort of bulk text input. And these things are shown here, kind of following a journal of a title, and you got an abstract and some keyword describing your experiment, link to publication, and your factors. So they're visualized in the web portal, as well as protocols, information about samples and assay. And these are eventually can be more structured maybe. I mean, there are other resources. For example, there's an NIH equivalent to MetaboLights called Metabolomics Workbench. There are more working towards the protocol acting as an SOPs. There's information about the samples and factors, assays, and metabolites that have been detected. Oops, sorry, will be reported, and you can see it. It's sort of a pop-up that you can link from Bio.js to KEBI. And you can actually, once you hover over that metabolite, it pops up the information about KEBI is displayed. A bit about reference layer. So there have, before was all the metabolomic experiments, but in a reference layer is focused on individual metabolites. So we pulling gradually from different resources, existing databases, people who have actually referenced chemical compounds in terms of a spectra or an NMR or a mass spec, gradually providing that to us or sharing it. And we enrich that by including that in the reference layer, information about the chemicals, chemical ID, such as species, what kind of reference compound it has, what of literature, what pathways has been known, and which study has been reported. So the MTBLS is our identifier. So information there, again, same structure of chemistry, some pathway. Again, this is another tool that was developed out of someone's PhD student to be able to look up different pathways that metabolite exist. We also look at RIA for reactions, Again, same BioJS, look up for one particular metabolite across EBI and get that information and show the reactions that these metabolites are involved, as well as being able to show the spectra compound. This is sort of how the NMR spectra of glycine betaine looks like. You can actually show different details that this spectra is being acquired. You can sort of overlap it. Again, this is a plugin tool that is written in JavaScript based on D3, and it can visualize different type of spectrum, from NMR to UV to IR to 2D NMR to mass spec. So it's a nice tool and shared, and there's a publication coming out for people to use and share if they wish to have this plugin to be able to use this to visualize their data. Some literature search, as well as other to browse taxonomy, for example. Getting to the data analysis side, so this is the analysis tab that I thought, and sort of being the next level, we have all these metadata and all these metabolites and the concentration, different levels of it captured. It will be good to provide tools to be able to do some statistical analysis or pathway analysis or some enrichment, other analysis. This is based on a tool, again, in collaboration with a group in Canada who originally developed this MetaboAnalyst tool. So they're happily shared it with us to be able to use it and make it available. It's an R-based data analysis tool. So beside to also, in addition to, I said mostly I'm making Cosmos. So this is sort of a standardization initiative, same 2012, primarily European partners. And again, this is sort of to get different people who are in informatics, improve databases or tools, or involved in larger initiatives and previous initiatives in standardizations or biobanks to agree on some sort of low-level or low-hanging fruit or top or bottom-to-top approach on agreements on how to capture the required metadata reporting. Primarily, one of the areas that is sort of emphasis in developing the exchange formats, the XML-based type of format that you don't depend on a vendor-specific format, but it's an open source XML, so you can actually use that for any tools that you have, and you can share it more freely and openly. Again, a lot of it initially started within the proteomic community in Ubuntu PSI, so we are trying to link as much as we can with them, joining their initiative and using the existing format and fill it up where actually there are missing. For example, there are tools in GCMS that primarily is not being used by proteomics community, so we're trying to actually include information that comes relevant to metabolite technologies that are not existent in or not used in proteomics, to include that, to add that, to work with vendors. So this is run by Stefan Neumann, who's mainly the guy who is dealing with this work package. So this is on several different levels. You try to actually use the ISO tab to capture this metadata, as well as linking it, having information on the raw files in an XML format that has all the experimental, all the details, technical part into it. So both of metadata and the raw information from instrument can be used for our databases and resources. As well as working, again, jointly with proteomics community, which started this format, MZ-Tab, to be able to include the small molecules into it. So this will be from metadata level to raw data and to what's been reported in terms of metabolite, how they identify the metabolite, how they calculated, how this information was quantified, and all the information relevant into IDing and measurements. Again, this is a large group of collaborators from people who make tools, softwares, and vendors or anyone that are interested in cross metabolomics and proteomics community. There are also a version for NMR that has been completely developed. There wasn't no equivalent XML type for NMR type of experiments, or nuclear magnetic resonance. Again, it's mainly used in small molecules, for example, although used in protein structure, but I'm only talking about NMR here. Follow the same format. We capture metadata in ISOTAP format and develop this NMR-ML. So we did a lot of work that was done in different consortiums scattered. We brought all together, be able to everyone who were previously developing different flavors of it to work together on one common format and be able to report, for now, for small molecules, how to an XML type of format, as well as same tab separated for IDs and quantification. And there's a website, nmrml.org, that is populated by the consortium and group, and you can go there in nmrml.org and see all of the information. It's about maybe 800 CV terms that's been added, a couple of comparators and validators that checks against MSI or Metabolomics Standard Initiative reporting that it complies or not. So this is just going, essentially this is quite new. We still haven't written the paper even for it, but we have the hard task now to convince people to use it. There's a lot of also joint workshop, I also said, with proteomics community to be able to bring this information on the tab separating format. So these are sort of examples of hackathons and workshops that we've done jointly or separately or partly by the Proteomics Standard Initiative. We're also working with journals. That was one of the things I'm missing, that sort of requirement to report metabolomics experiments is not mandatory. So essentially is gradually trying to enlist and get journals involved into this thing. But still, I guess you will see this in slides from others, and sort of the challenges they had, especially in file format, is the rate of data growth. This is sort of generally from EBI services, and you can see different services shown from Pride, MetaboLights, and how the data rate grows in terms of dotted lines being how much information doubles in a 12 month, and you can see metabolite is already surpassing. And if you consider the rate of the metabolite information that we have from metabolomic experiments, only makes really about 5% of actual available data or reported metabolomics data. You're only capturing very, very small percentage of the existing data, and still growing quite rapidly. So it's important to work on this format, on these compressions and new ways of doing it, and come to a decision how much data we want to capture, because if the rate goes by the same rate of growth, maybe it's not very sustainable in future to have all these big, large raw format data. We're also working on exchanging data and sustainability of resources within the NIH consortium. So this is a web shot of Metabolomics Workbench in the NIH-based initiative to how to get metabolomics data from people who are primarily NIH funded and make the data available. So we are actually trying to see how we can actually have some exchange of data in metadata level, in data level, and come up to some agreements on reporting. And one of these things end up in kind of a MetaboLicsExchange, pretty much based on some of the ideas that ProteomeXchange had. So I leave that for Juan to describe how MetaboLicsExchange was done, but this is sort of an equivalent version of it. But it's mainly is pulling information from different resources and bringing that information and sort of displaying how much data, for example, data set coming from metabolites, how much from Workbench. There are other smaller repositories, for example, Metabolomics repository in Bordeaux or Gaume in Germany that have shared the data and has some API access to this, all the metadata. Still, there are differences between how we capture a lot of this information. So there are inconsistency in the level of metadata that's been captured and reported, and so we're working to unify these things. Finally, the Cosmos Consortium, the ISOTAP team from Susanna, Felipe, Iman, and others metabolite team from software engineers, curations, and funders. So if there's any question, I'll be happy to answer.
Thank you, Reza. Yes, we have one question right now in the chat box, or several questions.
Should I scroll top? Okay, I haven't uploaded, sorry. I will upload it as soon as this is finished. Oh yes, so essentially it's a Lucene index which when these tab files which include all the information about investigation, protocols, publication, parameters, instruments, authors, anything else like that, it will be indexed, and you can actually look it up, but it has the limitation of what Lucene index has. So the ideal would be if we can move to more RDFs maybe, then we can actually have a better match in terms of looking up and linking information across studies. For example, if you're looking up for a study that runs on certain type of equipment with that particular column and has found its metabolites, then you will be able to find it. But for now, it's just a Lucene index.
Oh, thank you.
Starting metabolite database search, for example, I want to search for metabolic acid, and username is C181. When I looking for C181,
could you please first repeat the question?
Oh yes, I was trying to read and repeat. So there's a question about C181 that you're trying to look up, and when you look up C181 in a search from browse compound in the metabolite database, it didn't link me to oleic acid. Yeah, well, there is. It's not a very smart lookup for these things. There is using a Lucene index, I guess. People who work on this can explain this a bit more than I can do technically, why some of it is not going to be a very perfect match. But it's we're changing a bit of the architecture currently as well. We're trying to use a different architecture for the whole entire metabolomic repository. Before, we were very much relying on existing architecture, and what was done by ISA Creator and ISA tools. They primarily had this for a small lab, for someone individually could capture all this information, metadata, and keep it, store it, and show it, and you'd be used and look it up within their own group or small place. But for having it in such a larger and wider and more easier access, we sort of need to change a couple of things and adapt a new version, new methods of getting your data and metadata out of it. So we are actually in the process of changing some of these, how data has been handled in Metabolite, but it will take time for it to actually come up. Hopefully that will fix some of the issues. I don't think there's any more questions, or is it? In addition to NMR structure, do you also have mass spec? Yes. Maybe I've jumped it too quickly, but there is there is a mass bank database for small molecules and metabolites. Essentially, we are importing all the 16,000 different mass spec in there. So far, only different ways to do it, so we're trying to improve showing on different levels of fractionation and how much collision energy they use. So There are a lot of these things that we have to sort of download and figure out and unify between different resources that have information about reference mass spectrometry data and be able to do that. But you can already go and browse and see some of the entries that are from mass bank, and you can actually show different collision energy that are required and some of the metadata associated with that particular type of metabolite. So Henning asked about RDF. So no, we don't have yet any interface for it, for the RDF. But there are test versions of it on someone's computer, but not something that I can point you to. Sorry?
So what's the intended use case? So the idea is to use RDF as a sort of unifying format and eventually to allow queries across MetaboLights, for example, or even across multiple repository types. What's the perspective?
Oh, that's a good question.
I guess it's not just because you can.
Yes, it's a good question. Actually, we're going parallel ways. So, but one of the main routes we'd be thinking of would be the MetaboLights. So we have to sort of come to an agreement on how much information are fed currently. There's also differences of data that are being pulled by MetaboLights from MetaboLights and MetaboLights Workbench. There are differences in kind of metadata that we do. Ours is more enriched and has sort of CV terms and ontology. There isn't yet. So once, at least we can say from our part, we can actually do that. And the idea eventually would be via MetaboLights.
Okay, thanks.
So maybe I'll chime in there too, because I had a very similar comment as Henning, or question. I mean, I think, you know, EBI has certainly invested a lot of energy and time, and I think it's great in terms of making sure a lot of these data are available in RDF. And I think if the argument were solely data exchange between data repositories, that would be good, right? That would be a good application. But, you know, I think where maybe our center could really brainstorm would be sort of the data integration use cases that having all these resources exposed RDF would enable, right? These fundamentally enabling sort of analyses. And that's what I haven't seen sort of from any of the, you know, the semantic web community in biology. I don't know if you have any thoughts, or I'll just throw it out there.
It's a very difficult question. If I'd be the person who tried to just give an answer. I mean, there are always an issue, even cross-omics, of how do you interpret these data as well. I mean, there are sort of how meaningful the biology of it is for integrating data. I think there are first, maybe this is how I just gotta basically say what my feeling at the moment or little experience is, that maybe there are other challenges first we need to understand first, and how data cross-omics will actually work, or what kind of data, how do we interpret this information? Is at the biological level, the data level, and how meaningful these things are. And once we have some sort of good feeling and notion of understanding how these things work, then we can actually use this enriched metadata to be able to do that in more easier, accessible way. But again, I think this is generally a difficult question. We try to maybe tackle the easy ones for now within Metabolomics and within data exchange, and gradually as our understanding grows, then we move, at least from our part, towards more complex questions of integrated cross-omics data. Maybe this is not the best answer, but this is sort of gradually moving towards it. I don't know if anyone else have better comment.
So Henning's put in a few articles that I'll definitely go follow up on. But I think, this, I agree with you that there's sort of, within domain issues that need to be addressed. But I do think within our center, I mean, the big picture of things and the impacts on multi-omics analyses, impacts on cardiovascular data mining, things like that are things that we can and should be brainstorming. Maybe that's actually something we can, when we several of us are moving next week, that could also be an interesting discussion topic.
So we will probably discuss this in LA, but this pilot, which I mentioned before, which involves, which is anyway part of the planned work, between MetaboLomics, ProteomeXchange, and the genotype-phenotype archive. One where we want to build up exactly a metadata-based index. One of the sort of things I want to explore is if that would make sense to do it RDF-based. I have no idea if this will be the way we are going to pursue. It might very well be that we just do it based on XML and simple query based on XML, but I hope that we can at least explore whether the RDF would be the right way to go there.
Okay, there's one more question in the message box.
Okay, is that by Sarah?
Yeah.
Yes, that's me. But you've sort of answered it with this discussion. It's a little redundant. If we are short on time, we can move on. That's up to the organizers.
Yeah. Yeah, it's okay. So if anyone has any more questions. Okay. So if anyone has any more questions. Yeah, if anyone has any more questions, hopefully they can be answered after this presentation, or either next. We can have the discussion next week. But we'll move on to our second presentation.
Vincent?
Yeah.
A bit of echoing going on.
We do have time, right? You could have Sarah's question addressed.
Okay. I mean, I completely agree that, I mean, that's what mainly probably why we're here. I mean, we're interested in BD2Ks and centers like that to actually understand how things in a bigger question could be done. We have all these resources, essentially, that are present around here, all representing their own specific tools and application, and so perfectly good time, really, to see how things are going, how different resources and services are doing it, so we can actually plan ourselves. We are very, how could I put it, more a flexible service. Our entire data sets of metabolomics at the moment is about maybe 100, 200 different data sets. So if you want to change something, for example, is now would be a good time for us to change it and adopt it and make it available. Once we get into thousands of data sets, I mean, these are experiments, not data sets. The entire, which includes maybe thousands of little components, it will be too difficult or too time-consuming to change. But that's how I can sort of comment on that question. It's a good time, essentially, to be here.
Okay, thank you. Thank you. Right.
Are there any more questions? Okay. If not, thank you, Reza, for that presentation. We'll move on to our second presentation now. All right, so our second presentation. Is somebody here? Yep.
There's a terrible echo.
Yeah, I don't know.
I think the echo is from Juan, Antonio.
Really? But I'm not.
Is there?
I think if you mute yourself, I just see it showing up if you're talking.
Is it better now?
Yes, that's what's better.
I don't know exactly, to be honest.
Can you? Oh, that's a problem if you're. I guess maybe just use everybody else.
Fernando, you want to come?
I can go there if you want. I don't mind. Can try.
Do you have a headphone?
Headphones? Yes, I have.
Because then it avoid echoing.
Okay, I will go to the place and that's it. Sorry, 1 minute. I will go to Henny's office, basically.
Just want to use the opportunity. I mean, I have another meeting, unfortunately, at 5, so I'll be leaving at 5. If there's any question, just email. I will upload these as well into, or give it to Vincent to upload it into the Trello.
And we'll give you this presentation again on the long flight to LA, don't worry. Okay, so now we are going to try. I think you need to switch off your sound. We'll run the sound via mine.
Okay.
And then the optics go via yours. Switch off also your sound.
Yeah, but it is fine.
Now?
It's fine.
Okay, now you have to speak loud.
Okay.
Okay, so can you listen? Can you hear me?
Yeah, it sounds good.
Okay, good. So
I'll introduce the second presentation. So our second presentation will be presented by Juan Antonio Vizcaino. He has a master's in microbiology and a PhD in molecular biology. He has worked at EBI for 8 years and is currently the PRIDE project leader. Today Juan will present about ProteomeXchange, which provides a common framework and infrastructure for the cooperation of mass spectrometry-based proteomics resources by defining and implementing consistent, harmonized, user-friendly data deposition and exchange procedures among the members. He will also talk about the PRIDE archive, which is the data repository for MS-based proteomics data at EMBL-EBI. Without further ado, allow me to introduce Juan. Go ahead, Juan.
Juan, you're muted right now.
I think that was the idea, to use Henning's.
Henning's also muted, though.
Okay, everything's fine now. I unmuted myself, and so Juan will now not be muted anymore. We'll get there eventually.
okay, so now we are in the same office. Okay, so I'm just going to give you an introduction to Exchange and PRIDE. I'm going to talk about how the submission process to PRIDE works. I'm also going to introduce the concept of Proteome Central. So of course I don't have much time to talk about everything in high detail, but of course I'm open to questions later. Okay? So just a generic slide to start with. Basically, we all know that public availability of data in proteomics enables the interpretation of the results. So for instance, data reprocessing with different aims due to improved analysis software, change in protein sequence databases, which is clear in the case of proteogenomics studies, or the consideration of new post-translational modifications. On top of that, of course, it's useful for the validation of the experimental results reported, for instance, in publications. And of course, there are other specific use cases for proteomics, research related like, I don't know, spectral libraries, fragmentation models, or SRM transitions. So taking this in mind, okay, so now.
Something wrong?
Okay, yes, sorry, it was a bit slow on my side. So the ProteomeXchange consortium was started in 2006, but formally it was funded since 2011 by a grant of the European Union. And the goal of the consortium was to develop a framework to enable standard data submission and dissemination pipelines between the main existing proteomics repositories. It includes at present ProteoAtlas, the Institute for Systems Biology in Seattle, right here, of course, and Massive, which is very close to you, or some of you guys in San Diego. Two other repositories, Tranche and Peptidon, were initially included in the consortium, but as you know or may know, they were discontinued, so they didn't participate. They didn't participate in the first final workflow. So ProteomeXchange provides a common identifier space for proteomics datasets. It's what we call PXT identifiers. And at present, there are two fully supported data workflows, MSMS, TandemMS, and SRM, Selected Reaction Monitoring. And of course, our main objective is to make life easier for researchers. This is the URL, and all the details you can read in the publication that was published last year in Nature Biotechnology. This is how it works, the current version of the data workflow. So there are some components that are mandatory for each submission: the raw data, the processed results, and a set of metadata. This set of metadata was agreed between the ProteomeXchange partners. So this is basically how the workflow works. The submitter sends these data types to the receiving repositories. At present is Pride for MSMS data, Massive for MSMS data as well, and Puzzle for SRM data, and potentially other databases in the future, of course. The data remains private. We provide a username and password for reviewers and journal editors to check the data. And the data remains private during the manuscript review process. And then when the manuscript is published, or when the author tells us to do so, the data is made publicly available, and then all the information is sent to a resource that I will introduce later. It's called Proteome Central, and from that resource is basically made available to the whole community, to other repositories like ProteoAtlas and GPNDB that they could do reprocessing, or to UniProt and NextProt, for instance. Okay, so this is how the data workflow works. And I'm now going to focus, of course, on our work here at the EBI in the Pride database that is the main ProteomeXchange resource at the moment. So Pride stands for the Proteomics Identifications Database. It was started around 10 years ago by Leonard Martens, and stores mass spectrometry-based proteomics data, peptide and protein expression, identification and quantification, post-translational modifications, mass spectra, both raw data and peak lists, as much technical and biological metadata as possible, and at present, basically any other related information that may be interesting. And Pride, and this is important to highlight, has been focused from the very beginning in TandemMS approaches. Okay, it takes a bit to change the slides. I don't know why. Apologies.
Doesn't even change on your machine?
No. Maybe I have to do it this way. Okay, sorry. Again, it's some kind of technical issue. So Pride Archive is the Pride archival system since 1 year ago, January 2014. There are 3 iterations released so far, and it's still work in progress. I wanted to highlight that it was designed in a very flexible way and is developed development has happened in parallel with in the implementation of ProteomeXchange that I introduced before, but also with the new community PSI data standards. PSI stands for the Proteomics Standards Initiative, and Pride Archive system is based on these data standards, mainly NCTAP, that is the most recent one. It was published last year. MZML for peptide and protein identification, and MZML for MS data. So I don't have time to talk in detail about PSI standards, but basically Pride from the very beginning always has relied a lot in these community standards. Again, at the moment we rely mainly in MCTAP for identification and quantification. It was mentioned before by Reza. MZML for identification, and MZML for mass spectrometry information. There are 4 main ways to access the data in Pride Archive. I don't have time to talk in detail about all of them, but it's of course the Pride web interface, the file repository. We have a REST web service, and also we have a way to access information and visualize it. That is the Pride Inspector tool that I will mention later. So this is just a screenshot of the Pride web interface. I don't know here, I chose an example to look for data sets that contain metaproteomics information. This is just the result of a given search, and this is just another example of the web interface. I just wanted to highlight the protein identification page with the different peptides that match into the protein sequence. Okay, so the second section of this talk is devoted to how the submission process works and the different Pride tools that are involved in the process. So the Pride team is not only responsible of the Pride web interface, is also responsible for the development of all these tools. And first of all, I wanted to highlight the information that is needed to perform a ProteomeXchange submission. As I mentioned it before, but I will repeat it now. The first bit of information is the raw data. More accurately, we should call it mass spectrometer output files. It would be raw data, binary files coming from the different vendors, or pick list spectra in a standardized format, like MZML or MZXML. Then we have the result files, and for that we have 2 modes of submissions that I need to explain. One mode is what we call complete submissions. That means that the result files can be converted or are available in a standard format that we can read, RedXML or MZML, or partial submissions where basically we can take any data format. I will highlight again the difference here. For complete submissions, it will be possible for us to connect the spectra with the identification process results, and also these results can be visualized in either the web interface or in the Pride Inspector tool. However, in partial submissions, the process results are only available to download, but they cannot be visualized because basically they cannot be, they are not supported by Pride because we cannot pass them. However, at the level of the metadata, the way they look in the website, they look similar. However, at the essay level information in partial submissions is not so detailed. So it's important to understand this concept of complete and partial submissions. Again, complete submissions, it means that we can connect the mass spectra with the identification results. For partial submissions, we cannot do that. In both cases, raw data is stored and is available. Apart from raw data and result files, we need to have a set of metadata that it was agreed by the different ProteomeXchange partners. And then basically we have a collection of what we call at present other files. They are tagged in different ways, and this list can be extended. So at the moment there are quant for quantification related results, pick for pick list files, GIL for GIL images. There are FASTA for protein sequence databases, a spectral library for spectral libraries, and etc. There are a few more. I just explained the main ones. So there are 3 stages in each submission to Pride. The first stage is, and of course I'm going to explain here the complete submission mode. The first stage is the generation of the result files, And the way it used to work is that we were able to support the Pride XML format. That is basically the format that was used to develop Pride originally. And we had one tool that well, we still use it. That it was Pride Converter that made possible the conversion of some search engine output files from different software into the Pride XML result file. However, we are moving away from that, and what is very nice now is that the PSI standard MZ.ML for peptide and protein identifications is exported from more and more tools. So at the moment, there are tools like Mascots, Protein Pilot, Scaffold, PIX, MSGF, Peptide Shaker, and more. For instance, today we had some contact with Waters that are working on an export. So basically, the community is now adopting this standard more and more, and users, they don't have to convert files anymore, but in their software, it will be more common to have a native file export to MZ.ML. So this is the direction we are moving now. And again, this is just to highlight some of the tools that already support this export to MZ.ML. As I mentioned, Mascot, MSGF, PIX, Peptide Shaker, Scaffold, Protein Pilots, OpenMS as well, Mirimatch, and other tools from Dave Tapp's lab, etc. Apart from the MZ.ML, the reference spectral files need to be submitted as well. And in that respect, all open formats are supported, both XML and text-based. The second stage of the submission is the visualization of the results. Of course, this can also be done after submission, and for that, we have a nice tool that is called Pride Inspector. It was originally developed 3 or 4 years ago and published in Nature Biotechnology. It supported Pride XML and MZ.ML, and at the moment it also supports MZ and ML plus all types of spectra files, and also MCTAB for quantification results. These are just 2 screenshots of the tool. There's not the time to go into the details, but taking a look at the Pride Inspector can also be used as a visualization tool apart from the web interface. In fact, you can do many more things with Pride Inspector to visualize the data sets that are available in Pride. And the last stage of the submission process is the actual file transfer, and that is performed by the PX submission tool, ProteomeXchange submission tool. I didn't mention it before, but both Pride Inspector and the PX submission tool are open source, are developed in Java, so in principle they can be used in any operating system. This is the URL to download it. And what it does is it selects and captures the mappings between the different types of files, including a submission. So let's say a raw file is connected to a particular identification file or a quantification output file. And then at the end, it transfers all the files using the Aspera File Transfer Protocol, or FTP, if Aspera is not enabled in your institute. And also there is a command line alternative for people who don't want to use the GUI, for people who have, of course, bioinformatics support. I want to highlight here that, of course, raw files can be really big, and datasets can be also be very large. So that's why as this was also mentioned by Reza before, we are using the Aspera file transfer functionality. It means that, of course, depending on the location, can be up to 50 times faster than FTP. So file transfer speed should not be a problem anymore. Even from the States, we receive submissions, and they are done very well, very rapidly using the Aspera file transfer functionality. Of course, Aspera is also used in the command line functionality, and also now available for downloading files. I just wanted to finish this section saying that not only Pride doesn't only store MSMS data, which are around 90% of the data sets. Using the very flexible mechanism of partial submission, there are many other type of Proteomics workflows stored in Pride. Not SRM data, because all of it goes to Puzzle, but there are some top-down Proteomics data sets, some mass spectrometry imaging, or some data-independent acquisition techniques that are becoming more and more popular, like SWAT-MS or HD-MSC from Waters. So just want to highlight that Pride is mainly focused on MSMS, of course, but it can also store other types of data from proteomics for other proteomics workflows. Okay, so just the last section of the talk is devoted to Proteome Central and some submission stats. So this is the workflow that I saw at the beginning. So again, this is the submission process. The receiving repositories, the data remains private during the manuscript review process, but when the manuscript is published, the information is sent from the receiving repository to Proteome Central. And this is the resource that I'm going to explain now. Proteome Central is developed by our colleagues at ISB in Seattle. Basically, it's an interface for all ProteomeXchange datasets independently from the repository. So here you can see that in the same view, there are some datasets from Pride and some from Massive. And it allows search by metadata of all the datasets. So it's a place where you can go, this is the URL, and look for datasets that have been submitted to any of the ProteomeXchange repositories. Basically, it is based on the way we designed it. We thought that each repository, once a dataset was made publicly available, creates an XML format that was agreed between the partners and contains the basic information about the dataset. Basically, for you to decide if you are interested in knowing more or if you are interested in downloading the dataset. So it's, as I said, based on our XML format. And also something very nice, this is just an example for one dataset. So here you can see some details, like the publication, description of the dataset, etc. Again, you can use Proteome Central to search by metadata. And then something that also Proteome Central does, it has an RSS feed for announcement of public datasets. So you can get subscribed to this RSS feed. If you are interested, I can show you how later. And in an RSS feed reader or in an email, you can receive, now I think, daily updates for new datasets that are made public basically every day. And this service is, of course, open and available for everyone in the community. I just wanted to highlight that since mid-2012, when ProteomeXchange was started, the current data workflow, more than 1,600 datasets have been made available. At the moment, most of them, more than 90% of them are available in Pride. As you see, there are complete and partial submissions, and there are some submissions also available in Beta Talas Puzzle and in Massive. This data is from more or less 1 month ago. You can see that in 2014 alone, there were more datasets submitted than in the previous 2 years together, a bit more. And then at the moment, publicly accessible are more or less 50% of all of them. And this is, of course, because it takes some time, the manuscript review process. So 50% we think it's a very good number. And just to highlight that there are data coming from many different species. Of course, human is the main organism, but of course, other model organisms as well. Mouse, Saccharomyces, Arabidopsis, rats, E. coli, etc. USA is the country that has the highest number of datasets submitted, and the total volume of data in Pride at the moment is around 71 terabytes. The largest dataset submitted, and it was from the US actually, was 5 terabytes. And this shows that Aspera is really a good file transfer protocol to do this. I just wanted to, this is the penultimate slide, just wanted to highlight that this data is being reused more and more. I haven't included stats because I didn't want to run out of time, but it's clear that these datasets are being used more and more. And one example is the paper that was published in Nature last year by the Kuster group, when around 60% of the data used for the analysis comes from previous experiments, most of them stored in Pride, ProteomeXchange, Puzzle, or Massive. Okay, so just to conclude my talk, data submission and data reuse in the field are definitely rising. So you have seen that we are getting more and more datasets submitted to ProteomeXchange in general and to Pride in particular. Pride and ProteomeXchange enable this for you. ProteomeXchange is a collaborative effort, us here in the UK, but also by our American partners, ISB and Massive in San Diego. And also a key point is that data standards are really key for us. So without the development of MZML, NCTAP, and MZML, everything would be much more difficult. I want to acknowledge people in the Pride team, of course, here, but also our all ProteomeXchange partners, especially Eric Deutsch and Nuno Bandera. And of course here our partners, our funders. Yeah, thank you very much for your attention. I'm open to questions.
Thank you. We have one question. When you upload data to Pride, are you required to have process data like results from a search engine?
So yes, so I explained this before. So process results are always mandatory, but it can be provided. But there are two ways. What we call complete submission. It means that the process results are available in a standard format, MZML, or in the Pride XML, which is the format that we support from the very beginning, and they are always mandatory. So this is for complete, and for partial, we need also search engine output files or analysis your output files from your analysis software. So in both cases, either complete or partial, you need to provide some kind of process results. Raw data alone is not enough to have a ProteomeXchange submission.
Okay, thank you. We are considering for COPA-KB to let users, instead of uploading MZML from their local computer, take MZML directly, just give the accession number for something in the Pride database, and then search that data with COPA-KB, and then get a result file, and then save that file to the Pride data set.
The first part should be fairly straightforward and makes perfect sense. The second part, we can't directly modify submitted data. So if I understood you right, or is this only for those who own the data?
Well, this would be for, it would be for the person who, well, would there be a way for somebody to create, but no one can store the results without also storing the associated MZML, correct?
Yes.
Oh, okay, so
I think what you mean, or what makes sense, I think, is to allow somebody who is in the process of submitting to Pride or has just made a submission to then allow to use COPA-KB analysis to provide additional data, which sort of is then stored in their submission file. I think that's a good plan. What we can't do is we can't have third parties modify submissions from existing submitters. But I think that's probably not what you plan. I think you mean only people adding to their own data through the COPA-KB analysis, right?
Right. But currently somebody who uploads some data, so they upload just the MZML and they don't have like search engine results. Can they use the Aspera command line to upload data to continuously upload new pieces to a data set that's already uploaded?
So while they are private, there's a way to do it. Once they become public, at the moment we don't have a mechanism, but we are thinking to enable this mechanism this year.
Okay, thank you.
So I got a question from you there. Is there a minimum set of files that must be uploaded to a data set? Okay, so it's the same question.
The same question.
Are there any more questions from the audience? If not, we can end the presentations here. If anyone has any further questions, feel free to email me or Juan. If you email me, I'll send them over to Juan, or we can.
Before everybody goes, Pepe, are you on the line?
I am. Safe travel. We'll see all of you soon at LA.
So we are not going to, because you asked me something about chat, so we'll do that once we are in LA, right?
Yes. Could we do that, please?
Yeah, okay, perfect. Thank you. Sorry for this.
So, yeah. Or would you want to be online in another half an hour? I'll be in Skype.
I think we can do it once we are there, but I'll also be online in an hour. So if you want, then.
Okay. Oh, okay. All right. Yeah, I'm just I'm stuck in a little traffic. There's been an accident on the road. Yeah.
Oh dear. Good luck.
Okay. Thank you. Safe travel.
Okay.
Bye-bye.
I had a few questions for Andrew Su about GeneWiki, if you have time.
Yeah, sure. Go for it.
Yeah, so we're considering trying to expand some of the entries on GeneWiki, and there are a couple questions. One, I was wondering if we could basically build plain English sentences out of the gene ontologies and put those in the a function portion of the GeneWiki article. Do you think that would be valuable? Because it seems like a lot of the data, like the gene ontology, is already on the UniProt, but the purpose of having it on GeneWiki is to make it readable for people, make it easier to read. So do you think that would be useful to put the gene ontologies in sentence form, or would that be redundant?
I'm skeptical we could do it in a sort of a real natural way, right? They're listed on the right-hand side. I don't know if you noted there is a section on gene ontology in the info box. So the gene ontology things are there. To then list them out in some sort of formulaic thing of this protein is involved in DNA-dependent transcription. This protein is also involved in, metal ion binding, whatever.
Right.
Over and over again. I think that would not be, I don't think it would end up being a very readable paragraph. Or did you have something else in mind?
That was sort of it. We're just trying to come up with ways to do it elegantly that didn't just seem like a large list that was hard to read. Do you think there's room for much of the text, like in the function portion of the article, to be written by a machine, or do you think that has to be something that has to be hand done?
My sense is that has to be hand done. I mean, there are some proteins with, I don't know, 30, 50 gene ontology terms, and again, to just sort of, reformat that, and I actually think it's easier in the info box where you can sort of scan through it. I mean, look, I don't categorically say it's not worth doing, but if you want to, take 2 or 3 proteins and just sketch out a few ideas and then circulate them, I think that's worth all of us looking at.
Okay.
Does that make sense?
I think it would need to be very clearly marked as auto-generated, and from what? Because if you generate text, then this kind of test is human done, and I think this would be very potentially misleading to have, let's say, Go terms which originally have been created by some automated process, then you turn them into text and it looks like they have been added by a human being. I think it would be need to be very clearly marked.
Mm-hmm.
Okay, could I intervene here? So I think, Brian, maybe we gave them a bit more background of the questions where they came about. What our group is interested is to actually taking care of 2 specific subsets of proteins. One is any proteins that involve the cardiac cell contraction, contractile functions, and Dr. Sarah Scrax is leading that effort. The other set is looking at all the core mitochondrial proteins. This is about 450 proteins that's been shared by 5 different mitochondrial model systems. There's different species or organelles. So not the 2,000 mitochondrial proteome list, but the core, the commonly shared mitochondrial proteins. What we found by reviewing these 450 mitochondrial proteins online is majority of them have had info box on the right-hand side, but the main text is one sentence. It's just a, well, this is a gene of this gene name, and you can backtrack it to UniPro or gene ontology, whatever that is. And there's nothing else. So it's very simple. It looks to us it's a machine-generated sentence, and we could be totally wrong. So this is actually a discussion point that, as Brian pointed out, and Anders and Brian and Jennifer and a number of them working on the mitochondria project have been facing. So we would welcome any advice, recommendations, Andrew, from you or from others who are familiar with the topic, because they're definitely poorly annotated GeneWiki. These are just like one sentence, nothing else.
Yeah, no, there's definitely plenty of those, and those, you know, those are these quote-unquote stubs.
Right. So with the expertise our group has had in the past 15 years studying mitochondrial proteome, so those for us look like the entry point if we want to be of assistance or to make a contribution to GeneWiki.
So, you know, I think sentences that span multiple proteins that, you know, so for example, you know, gene A, gene B, gene C are part of a complex that participates in blah, 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 blah. And if you wanted to put that sort of, you know, very short paragraph on all, you know, gene A, gene B, gene C, that I think would be perfectly fine and great.
Otherwise, you would recommend a carefully constructed functional analysis as text.
Yeah, you know, I just worry about the auto-generated.
Right. Right.
Yeah. So human-generated and copied.
It's also difficult. I'm sorry. The auto-generated thing is also difficult for future edits to take place, right?
Oh yeah, you do it once and you're done, and because it's very difficult to differentiate if somebody else has come in and tweaked it, and the last thing you want to do is have a bot overwrite what a human did.
Right, right. So if we would come in, write the text, and what I've seen that's very eloquently done there is, you know how some of the words in the text are also linking to other pages. Are there technical things we need to be aware of when we construct things in sentences like that?
Sorry, to set up the hyperlinks to other pages?
Right.
Yeah, there's a very, very simple syntax there that it's actually just a double square bracket. Usually if you enclose the word in a double square bracket, usually it'll link correctly.
Okay.
I think it would be useful, you know, just so you guys don't find yourself running in circles. Like if you wanted to sort of work out the first few of these iteratively with me or somebody in my group,
yes.
I think it would be useful.
Yes. Okay. Okay. So that's what we're looking at. I think Sarah is looking at a couple of proteins from the contractile function family, and on this end, Anders, Brian, and Jennifer are looking at a couple of starting points for the mitochondrial core proteome.
Okay, great.
And Andrew, I have one other question. How do you determine the notability for a gene or a protein to create the article?
At this point, we are done doing mass creations. You know, we did like 9,000 or 10,000 of these, just sort of ranked by the most number of linked publications, and that was just sort of agreed on by the community. But at this point, right, if any individual person wants to create a new article, I doubt you would hit any resistance in the community on sort of a one-off or a family sort of based effort. As long as there was some efforts to put in something beyond just the stub that we auto create.
Okay, thank you.
Yeah, definitely. Let me know if I can help or offer thoughts on.
Yeah, would you like to speak to our group that's doing because you'll be at UCLA on Monday, correct?
Correct.
Okay. Yeah. Maybe you would like to meet with Jennifer and Anders and those working on the GeneWiki.
Yeah, sounds good. I have no idea what else is planned for Monday afternoon. We're going to show up and be overwhelmed by how much we have to coordinate. I do want to make sure we coordinate as much as possible with Henning and the European team because they're geographically much more distant. There'll be more opportunities for LA and SD to coordinate.
Yeah, absolutely.
Great.
That sounds good.
All right. Well, thank you everybody for attending. We will stay on the line till 10 o'clock if there are any further questions. But that concludes the presentations. Thank you.
Thank you. I'll be on Skype in 5 minutes. Thank you.